Germany. And Becca, who was previously a graduate student at Georgia Tech with Lauren Williams, has now taken an instructor position at Queen's University of Charlotte. So we want to sincerely thank both of them for keeping the show running for 3 years. And we wish them all the best in their further endeavors. So I would like to introduce the topic for today. So in the seminar series in general, what we're ultimately trying to figure out, of course, is how did life on Earth begin? That's our grand unifying interest. And today we have a speaker who's going to give a particularly salient presentation on that topic. And I thought I might give a conceptual overview of what it is that we're trying to solve. So, let's see. I've prepared this, and it is what I'm calling the singularity of terrestrial biochemistry. What I mean by that is that life on Earth is of only one variety. We have only one example of life. And it is this one example of life that we have extrapolated pretty much everything in the top-down manner on how life on Earth could have began. And this is an overview of what it is we're really trying to explain. So, of course, we're all familiar with DNA, the hereditary molecule of all life. Now, I suspect that most of us are also familiar with RNA, which is a molecule that functions similarly. Together, these form the nucleic acids. Now, in all life on Earth, we have this substructure of DNA and RNA, and of course, they're quite related. Then there's another class of polymers called protein, which are quite distinct from DNA and RNA, and yet they are intrinsically linked by a flow of information. So first we have the flow of information from DNA to RNA. That process is called transcription. Then we have the translation of an RNA code to a protein sequence. Now, we can also have the flow of information back from RNA to DNA. That's called reverse transcription. And then finally, there is the replication of DNA, which propagates all life on Earth. This concept, this flow of information in biological systems, was given the title of the central dogma of molecular biology by Francis Crick. And really, this is the thing that we're trying to solve, in my opinion. This flow of information between disparate classes of molecules is the ultimate mystery of the origin of life. And so, as probably all of you know, the main attempt to explain how this self-referential system could have come into existence is via RNA, because RNA plays both informational and catalytic roles in this process. Still, though, that doesn't explain how the entire informational system came into existence. And our speaker today is going to give us some insight on how the beginnings of that may have occurred. So I'm going to yield the floor to James. And James, if you wouldn't mind pulling up that slide. So, okay. Thank you. So our speaker today is Dr. Sam Roberts, who is a postdoctoral researcher in the lab of John Sutherland at Cambridge. I'll not read straight from the slide, but what I'd like to emphasize is Sam's expertise in this field, as he's been working in it for quite some time. And I'm very much looking forward to his talk, which is titled RNA Aminoacylation at the Origins of Life. So Sam, if you'd like to take it away, we're very happy to have you, and we're very excited to hear your talk.
Thank you very much for the kind introduction, and yeah, a great kind of preview on some of the stuff that I'm wanting to talk about. I think screen sharing is currently disabled for me, by the way, so I can't show any pictures, so you'll just have to look at my face for a couple of seconds while we sort that out. But yeah, no, it's David's. That was a he's almost stolen my thunder a little bit uh, with the first couple of slides. So you'll have to forgive me if there's a couple of um, uh, cut if I repeat myself a little bit. But yeah, so what I want to talk about is our kind of efforts towards understanding where the genetic code comes from and the processes that encode for proteins and how that relationship between protein identity and genetic code essentially came from. And also put it in the wider context of kind of John's or the scenario that we're invoking, which is kind of mostly from John's, but others work as well. And I realize this is a fairly broad kind of audience, so I'll be trying to kind of approach this in a manner that hopefully everybody can follow. So yeah, sorry if I make things a little bit simple for some of the members of the audience, but hopefully it'll be engaging. So as David rightly said, the kind of our real focus is this central dogma of molecular biology coined by Francis Crick lately of this parish and Nobel Prize winner. And this is interesting essentially because of its commonality, right? You know, thinking about how diverse you know, the tree of life is. All modern life has this kind of has these three molecules in common and their relationships in common. And for many scientists over decades, the question is how could this system have appeared? And the analogy I like to use to try and think about the role of these three molecules is essentially if you think of DNA as like the zeros and ones on a computer hard drive, and proteins broadly as the outputs of a computer, right? You know, the screens that we're looking at now, or the speakers, or the printouts from your printer. RNA is essentially the software molecule that's changing that one form of information into another. And but much like that kind of oversimplified analogy, this system is way more complicated than just this diagram kind of suggests, right? And it's largely been considered that this system, as it stands, is way too complicated to have formed kind of spontaneously as it is. And so the question has been, what preceded this? What preceded the modern central dogma of biology? And usually the question has kind of basically been reduced to which of these three components came first, because essentially essentially, as David mentioned, the one that's kind of won out has been RNA, because as well as its kind of modern software type role, it can also store information. So we're all too familiar with COVID-19 and other viruses that use RNA as a genetic store. But RNA can also be a functional molecule. So RNA can fold and create catalysts, just like proteins can. And the most important one, I guess, being the ribosome, which is its central core is primarily RNA, and is actually a machine we'll be talking about later on with respect to building proteins. And so the idea came that perhaps actually the first forms of life were actually purely RNA, and hence the title, The RNA World, which I think most of us are probably familiar with. Not to say that this is a kind of a starting point for how I want to talk, how I want to present the information. It's not necessarily exactly how we believe life started, but it's a familiar theory that is a useful starting point. And if RNA is to be central to the earliest forms of life, and if we think it's one of the earliest biopolymers, then there must have been a prebiotic synthesis of it, because at the moment in modern biology, RNA is constructed by proteins and through all complex processes that wouldn't have been available at the origins of life. So the question has always been, where does RNA come from? I'm not going to go into loads of detail about this because I want to get on to the kind of aminoacylation stuff, but I figured I would give a brief overview of our kind of cutting edge hypothesis of where RNA would have come from. But before we do that, we have to understand what RNA looks like. And I realize to non-chemists, this may look scary. But essentially, you don't have to be a chemist to kind of realize that this is a really complex molecule, right? So you have loads of different ring sizes, lots of different elements. You have bonds in specific directions. So, for instance, this bold bond here means that it's essentially pointing out of the page. It's the kind of way that we represent, 3D molecule on a 2D page. And there's also certain particularities. So you have this repeating backbone chain of phosphates and ribose, and you also have the bases here, which are which make up the letters that we're kind of used to seeing. So we've got U, G, C, and A here, and they don't need to be in a specific order. They're any order is or even subset of this is still called RNA. And in fact, this when we're talking about RNA as a genetic molecule, yeah, the genetics or the particular information is defined by the order of these bases, right? So we essentially the convention is to read from the top of this molecule down, as I've drawn it, and then to kind of essentially the definitions of the letters defines what the information is stored in RNA. But before we kind of think about how RNA could be useful at the origins of life, like I said, first we need to know how it's produced, which has been a long-standing challenge that John has aggressively tried to solve or provide a solution to. And we think that the key molecule, key feedstock on early Earth to do this would have been hydrogen cyanide. That's because hydrogen cyanide could be made in large amounts in the early atmosphere, either during lightning strikes, or we actually think probably the most likely source of large amounts of hydrogen cyanide would be large meteorite impactors, breaking nitrogen and carbon dioxide, and then reforming to make hydrogen cyanide, which can then rain out of the atmosphere, and water as well. Of course, you need water. And then we can collect hydrogen cyanide in salts, things like ferrocyanide and so on like this. Cyanide's also particularly attractive because of we have carbons and nitrogens, which we need lots of for the nuclear bases, but it's also very active. This triple bond is kind of asking to react and make things. So once we collect this hydrogen cyanide, a series of steps get us to this molecule, RAO. And I noticed Furkan is in the audience, so I'm going to plug some of his work for a little bit. And the reason that I've kind of pointed to this molecule is that it's essentially a node in various chemistries and particularly the synthesis of the different nucleotides in RNA. In that after this point, things can start to diverge. It's also a crystalline molecule. So whilst there are various steps that, even though they're quite efficient, will inevitably make various byproducts, this molecule will crystallize out a solution, and then all the kind of remaining material can be washed away. So this is one of the rare instances where you can essentially do prebiotic purification, right? And what's particularly exciting and why I want to shout out Furkan is because he's found out that this crystallization can go in a enantiomer pure manner. So this requires me to talk about what enantiomer pure means. So step back a little bit. If you take your two hands, they are non-superimposable mirror images of each other. So there's no way that you can overlap your hands and make them look identical, right? You've got left and right. And this is a concept that is kind of universal to kind of various structures and is true of chemistry as well. And so this molecule that I've drawn here, RAO, you could easily draw the mirror image and kind of and it would be a non-superimposable molecule. What's relevant, why this is relevant to the origins of life, is that biology is also chiral. So I would say the vast majority of macromolecules and small molecules actually in biology are also themselves chiral. So the nucleotides in DNA and RNA all have this chiral labeled D, and amino acids which are the building blocks of proteins, are all L. And so there's this question of how did this chiral imbalance occur? Because conventional organic synthesis should produce both enantiomers. And so there's been a long-standing question about where this breaking of mirror symmetry occurred. And people like Donna Blackman, she's had a recent paper in Nature, who have spent a long time thinking about this problem. And I think this work by Furkan is just a brilliant example and probably the most compelling potential mechanism for doing this. And essentially, it invokes having magnetite, which is a mineral that we think is immensely prebiologically plausible. And when that interacts with the magnetic field of the Earth, it can itself magnetize, and through a very fancy physics effect called the Cis effect, which I'm definitely not an expert in, but essentially this you can enable chiral crystallization of molecules on this magnetized surface. So essentially, we can, with this crystallization chemistry and invoking magnetized magnetite, you can purify one enantiomer or one mirror image of this molecule RAO. And this chirality will be maintained through to the resultant RNA molecule. Subsequent chemistry, which is kind of dependent on particularly various sulfide molecules that can be emitted from volcanic outgassing, think like the geysers at Yellowstone and so on, but also crucially UV light allows us to get to this molecule here. And so this means that we can start to put certain constraints on the type of chemical environments that allows us to get to nucleosides, which are crucial to make RNA. So all we need to do now is add phosphate and couple these two things together, both of which we can do by wet-dry cycling on minerals containing phosphate. And essentially, there are still some gaps in this story, particularly the purines are still something that we have roots to, but we're not particularly satisfied with. But there's some really exciting work going on in the lab at the moment about. But I think actually the real killer, which I know Dieter has been working quite a lot on in particular, is about this oligomerization step. Fighting entropy to build long oligomers is really hard and still something that I think is a really interesting problem that needs work on. But I think hopefully this has kind of provided a background to kind of say that we think that RNA is a plausible molecule on early Earth, and that we think that we have a decent route to it on the surface of the Earth in kind of shallow pools that can interact with UV light. So now that we have RNA, we're now thinking about that RNA on its own isn't life, right? Even if groups like Phil's and Jerry Joyce's managed to make self-replicating RNA, there's still a question about that I don't think certainly a chemist wouldn't call that alive. So we're trying to think about how what else is kind of required, and in particular the other elements of the central dogma. And proteins are kind of where we're turning our attention to. So proteins are biopolymers that are actually conceptually made up of kind of simpler building blocks, right? These amino acids, which are only 3 atoms long, but their diversity in side chains allows the kind of complex biochemistry of proteins to occur. And potentially you could build, much like we've just built random RNA in the previous slide, we could build random peptides or proteins. And I've highlighted a couple of papers from my PhD lab, which give a couple of really nice chemistries of how to access these in a completely random manner. And proteins are also interesting from our point of view in our scenario that we're invoking, because essentially the same school of chemistry that allows us to get to the nucleotides, so nucleotide compound 10 here, we have actually arabino, but the ribo oxazoline that I showed earlier, compound 5 here. The same chemistry allows us also to get to at least a subset of the amino acids that are present on the early Earth, and these are all ones that we think are plausible to have appeared early. There's this idea of some amino acids appearing earlier in the chemical record than others. But crucially, these amino acids are synthesized in an achiral manner, so that we still have both mirror images. We still have both enantiomers in these syntheses, because Furkan's work happens later on than the syntheses of these amino acids. Right, the divergent point to amino acids is earlier than RAO. there's also a conceptual problem about randomly making proteins, and that's a kind of statistical one. So um, I'm sure there are people who could come up with better numbers than these, but a very quick Google tells me that the average protein length is something like 300 residues long, and there's 20 different natural amino acids, and conceivably there could be a lot more unnatural ones that could be produced by prebiotic chemistry. And so when you start to kind of generate all the different options that you could make from these kind of vast set of amino acids, you get to add like really astronomical numbers of different proteins. So even if if there's a particular protein that you want, or even a particular family of proteins that you want that have some kind of activity, say to stabilize RNA to hydrolysis or improve its replication capability, say, then you're there's still going, and you're planning on making these proteins randomly, you're still going to make them in the context of a lot of other noise and a lot of other prebiotic clutter. And so this is something that we kind of try and need to think about how to avoid. Biology would also have this problem, and it avoids this by using translation or using the genetic code. So essentially, it uses the molecular machine, the ribosome, to read the genetic code, to read the instructions for the particular protein it wants, and then from those instructions, it can sequentially kind of add, the first amino acid, the second amino acid, third, and so on, and then fold this kind of mature protein and stop, so that it doesn't keep making a longer, forever longer and longer protein. And so it's essentially leveraging a relationship between amino acid identity and genetic code to avoid this clutter problem and also to have control over what proteins it builds when. And this mechanism, again, is universal to all of life and is kind of essential, and we think is very, very ancient. And so we're interested about where this comes from. And this finally actually gets to the subject of my talk, RNA aminoacylation, because we want to understand where this comes from. And to start doing this, we've looked at tRNAs, and particularly aminoacylated tRNAs. So this is a tRNA molecule, which is an RNA chain that's 78 bases long, so pretty long, and quite complicated when you kind of look at it just like this. But essentially, I think of it as a kind of bifunctional molecule. You have at one end, like a decoding region here. So this is 3 bases that correspond to, a 3-base region of the genetic code, a specific 3-base sequence. And at the other end, you have the corresponding amino acid. And so this is a kind of a bifunctional molecule in kind of chemistry terms. And I kind of compare this to like an entry in a language-to-language dictionary, right? So you have you're going from the language of the genetic code to the language of amino acids, and this is the entry that says that the word hello is bonjour in French, essentially. And so it's essential that this relationship is maintained, right? So that, I mean, there's a Monty Python sketch about somebody who deliberately makes a Hungarian phrase book that's kind of nonsense, right? And so ensuring that the relationship between genetic code and amino acid is maintained is important, because if they get scrambled, then the genetic code is no longer, at best, no longer a code, or worse, kind of actively malicious, right? It's actively making things that you don't want. So, and biology maintains this relationship with tRNA aminoacyl synthetases, these enzymes here. And this is the chemistry it uses. So it takes ATP and an amino acid, makes a bond called a mixed anhydride functional group here that I'll talk about, and then loads within the active site of the enzyme, loads it onto the orange region of the tRNA. So I've kind of truncated the tRNA in this diagram here. The protein itself, or the enzyme itself, is a protein, and so is also a function or product of translation. So when we're trying to think about where this relationship could have come from prebiotically, there's this kind of chicken and egg problem, right? That you need the enzyme to ensure the relationship, but to make the enzyme, you need the relationship to already exist. So this enzyme couldn't have already existed, and so we were thinking about what chemistries could have existed to allow this aminoacylation prebiotically. To do this, we've kind of truncated the tRNA to just this purple and orange region that I've already kind of talked about, and I've kept the the kind of biosynthetic pathway here just for reference. We've got two different chemistries now for how this could have happened prebiotically, and the first is essentially leveraging this idea of a mixed anhydride bond, but rather than it being on an adenosine free adenosine molecule, we're actually using the power of intramolecular chemistry, essentially, or templating. Excuse me. So there's in modern day tRNAs, there's always a 5' phosphate at the terminus here. So if we leverage that phosphate again with a mixed anhydride bond, this orange arm overhang can essentially reach around and pick off the amino acid from here, making the same product that biology would produce. And this is kind of cool because there's a kind of question why are tRNAs phosphorylated, because there's not an obvious reason why they need to be phosphorylated in biology. And so maybe this phosphate is actually just a kind of a relic from the prebiotic chemistry, essentially. However, the how you make this bond prebiotically is not satisfactorily answered in our opinion. So when I joined the lab, we also developed another chemistry that essentially does the same thing, but flips the amino acid the other way around. So we actually start from a phosphoramidate bond, and then through 2 steps of activation and hydrolysis, we can get to the same product. And we think that this is probably a bit more prebiotically plausible, but we're interested in both chemistries still. So given these two chemistries, we're kind of interested in looking and studying what happens when we start varying things. And in particular, what we're interested in is going back to this idea of stereochemistry. So thanks to Furkan's work, we know that we should have enantiomer pure or one mirror image of the RNA. And that's what we've just been using the natural L RNA for all our studies because it's the kind of cheapest and most widely available RNA. And what we find is that if we look at the 2 different mirror images of amino acids that we can kind of couple with with these systems, we find that both of them actually have a preference for D amino acid. Sorry, for L amino acids. So we're starting from, I apologize if I've got this wrong. Definitely starting from dRNA. And dRNA likes to prefers to transfer L amino acids kind of across the board. So, and this is for both chemistries. So you can see here that we transfer the L amino acid, and you can see here for the second chemistry we also prefer to transfer the L amino acids. And this is really cool because this is saying that with a mixture of amino acids, and the currently natural RNA, we preferentially transfer the natural amino acids. And so this sets up this relationship between between LRNA and between dRNA and L amino acids. And crucially, it means that we don't have to invent chirality twice. Once you've invented it in RNA imprints on that chirality onto amino acids. And this would be important because not only is biology homochiral in its proteins, but if you had a mixture of the different enantiomers of amino acids, then that gives a whole new dimension of the complexity of making proteins, and most of them again would be useless and inactive. So given this kind of really cool stereochemical effect that we see with both these chemistries, we're now interested to see what happens if we vary the sequence of RNA, because I said before that you can essentially change the identity or the order of the different bases, and provided that you can form the same structures, say the same base pairing, it's still the same kind of overall structure. So to do this, we essentially took a fixed stem sequence here, so UCGCU, and the complementary sequence opposite it to form a duplex, but then had a library of randomized sequences in this overhang region here. And then through the magic of next generation sequencing, essentially we were able to identify which sequences transferred amino acids better. And we did this for a variety of amino acids and came up with this kind of rather complicated set of data, which I'm going to quickly try and talk you through. because there's various different ways of looking at this, but they will come out with the same answer. So I'm going to start off. I think the easiest one is to look at the diagonals here. So what these represent are essentially the sum of all the sequences that come up in the top 10% of reads. So it's the top 10% hits that come out of this. So and essentially so we have 5 columns, and they correspond to the 5 different bases in this overhang here. And essentially, the bigger the letter, the more prevalent that base is at that position. So if we look at the L-alanine column here, the first position, N here, is mostly uridine, for instance. And you can do this for all the cases. And when you look across all the different amino acids, we find that generally these motifs look very similar. They're kind of very UG rich, right? For this, and this is just for this table here is for chemistry 1, for the anhydride chemistry. And crucially, that this kind of generalized motif is different to the background. Another way of looking at it is the correlation. So what we're saying by correlating is comparing 2 amino acids, say glycine and alanine, and a correlation of 1 would say that basically all the sequences kind of transfer glycine and alanine equally. So a sequence that transfers glycine well also transfers alanine well. So closer to 1 means that you have that there's kind of no difference between the amino acids. And this correlation is demonstrated pictorially with these graph relationships here. And you can see here that with both chemistries, essentially, although they have different preferences, for overhang, there's basically limited differences between the different amino acids for preference of transfer. And it's not surprising that they have that between the 2 chemistries are different, because the orientation of attack is different in each case. But yeah, we don't see any difference between the amino acids. Things get really exciting though when we start looking at changing the stem here. So we again have this time we fix the overhang, but have a variety of different or a library of different sequences in the stem region here. But we also provide the kind of complementary sequences in the library here. So then we always have this kind of duplex phenomenon. And whilst unfortunately the chemistry that I was working on again shows a kind of essentially a rising tide lifts all boats, that there's this AGU is the kind of winning sequence for across all amino acids. When we look at the mixed anhydride chemistry, we now see vastly different sequence preferences for different amino acids. So the sequence that transfers L-alanine is very different to the sequence that likes to transfer L-proline. And this is really cool, because this is the first evidence of a prebiotic enzyme-free relationship between the sequence of RNA and the amino acid identity that we're loading on said RNA. Sequencing, or these kind of readouts, are very kind of abstract and not a kind of a chemist's idea of yields. We're kind of using reads as a surrogate for yield. So we wanted to kind of test this in a way that's kind of more familiar to us, and also to validate the sequencing, because yeah. And to do this, my colleague Meng, who also did loads of the sequencing, and I should thank him for letting me show loads of his data today. We used an HPLC assay where essentially we took we took the results from the sequencing, synthesized the strands that correspond to those results, and then loaded a set of the amino acids onto each of the sequences and kind of followed how well they transfer. So for instance, in A here, we've taken the base sequence for L-alanine and shown that it transfers very well, topping out at 60%, but also loaded the same other amino acids onto the same sequence and shown that they don't transfer as well. And then we've done it for the other amino acids as well here. And there's lots of kind of nuances that you can look at all this data. So the selectivity between L and D alanine varies. So for instance, in this case, where the leucine transfers very well, actually the L and D alanine transfer kind of actually quite equally, but they are the minor product, or would be the minor products. There's also differences in stability. So for instance, it's known that valine has valine amino acyl RNAs have very long half-lives. And there's also kind of issues with glycine here that glycine is always one that struggles, but we can imagine that glycine might be around a lot. So this would only be this kind of level of selectivity is only going to be one part of the story, right? If you're making all the different amino acids in different levels, that's also going to then stack on top of this transfer chemistry. And there may be other selectivities that all kind of culminate in the kind of total amount of each of the RNAs that are around. But essentially, this data set validates the sequencing that there is selectivity to be found here. So let's zoom out back to the tRNAs. What have we actually found? When you're thinking about how the identity of the amino acid might have been linked to the genetic code, you would naturally think, oh, it must be something to do with this sequence here, the readout of the tRNA, or the anticodon it's called, that reads the genetic code. There must be a relationship here. But the problem is here, this distance is 75 angstroms. It's an enormous distance chemically, and it's really hard to see how an amino acid could essentially be affected by this region. And what we've actually found is that the amino acid, the identity of this amino acid is actually determined by the last three bases of this purple region here. And this is really interesting because when we look at modern day tRNA aminoacyl synthetases, they also use a variety of different determinants, but a lot of them actually read this region of the tRNA to determine which amino acid to load onto this molecule. So this kind of suggests a scenario where if we're right with our chemistry or something similar to it, aminoacylation happens in this kind of semi-coded fashion, because of course, this isn't clean coding. We're talking a factor of 2, 3, maybe at best. And obviously changes over time as well. But there is selectivity in the aminoacylation, the aminoacyl transfer, and then the kind of nascent aminoacyl synthetases pick up on this selectivity and amplify it, and they pick up that it's the purple region of the RNA that matters. And then later on, there's a transition to also recognizing other bits of the tRNA, including the anticodon, as these enzymes develop. So this is only one kind of part of the tRNA, and we've also interested in various other aspects. We've got a couple of interesting papers that are under review at the moment, and both are on Bio and Chem Archive, including some work by my colleague Mung, who's been interested in which anticodon-codon interactions, so which binding regions or reading regions of the genetic code would have been most important early on. And we're also interested in starting to make peptide bonds and what the actual origin of translation could have been. And just to finish off, I just thought first I'd like to thank all my colleagues present and past, particularly John for his great mentorship, and Mung and Ziwei Liu, who's an ex-member of the group, who've kindly let me show a lot of their data today. And all my previous funders and everybody. But what I wanted to finish on was just a kind of direction of travel for how we're thinking about the origin of translation. So because there's this problem that making proteins is a very complex process, and kind of it's not clear how translation could have got started. Or it's a lot of steps for there to be any evolutionary payoff, essentially. Because you essentially, for evolution to kick in, you either need proteins to be stabilizing RNA or improving their replication capabilities, right? So we're trying to think about how could what would have been the stepping stones to translation evolutionary. So in a scenario where you have some kind of replication mechanism, right, there's various that you could invoke. You essentially what you're taking is an RNA sequence, which I've kind of this is RNA, but I've redrawn it as this kind of simplified diagram here. Essentially, you're loading on the next base, right, uridine here, and this is via some kind of activation chemistry. But if you're doing this activation in the presence of amino acids, you're also likely to aminoacylate, perhaps by one of the chemistries that I've been talking about today. But once you have that amino acid group on there, this prevents you from replicating further. In chemistry parlance, you call it a protecting group, right? It's blocking, it's stopping this process. So we have to find a way of removing amino acids. Of course, you could just wait and let this bond hydrolyze because this is hydrolytically labile. But if you have long RNAs, and depending on the identity of this amino acid, you may actually suffer from getting significant RNA hydrolysis on that same kind of timescale. So there would be an evolutionary pressure for developing a mechanism for deacylation. And one way to do this would be through translation, because if you had two of these molecules next to each other in the right conformation to form a peptide bond, you go from two blocked RNAs to one blocked RNA, where we now have a RNA with a dipeptide on it, and we now have our free RNA that can now replicate. So essentially, we have this sacrificial RNA that's going to take the hit for the team and produce dipeptidyl RNA. That's essentially a waste product in this process. So our idea is that essentially we're using that replication is the driving force and would inevitably make peptidyl, dipeptidyl, and this could keep going, to try, and longer and longer peptides, but they're essentially a byproduct at this point. And then later stages of biology would have then found, oh, we've found this way, we've been making all these bonds, and it makes all this cool chemistry, and we have this kind of selectivity. Let's see if we can exact this. So using exactation, maybe we could then build, kind of translation from there. And this is kind of how we're thinking about the evolutionary processes. And I'm sure this will maybe spark a lot of debate. But I've been talking for far too long at this point. So I'll just thank you all for listening and welcome any questions that you have.
Sam, thank you very much for an excellent presentation. That was very interesting. So I forgot to mention at the beginning of the seminar that obviously we'll hold a Q&A session right now. I think since there are a handful of questions in the chat, we'll get to those first, and then I see that there are a few people who have their hands up, and what you can do is just unmute when it's your turn and ask your question aloud. So one of the first questions I see in the chat here, Sam, I think you probably addressed this, but I'll read it anyway. Manon asks, the pattern of coded acylation, is it stochastic or controlled and deliberate due to environmental factors?
Is it stochastic or controlled and deliberate due to environmental factors? So we don't have a good mechanistic explanation, which I think is what you're trying to get at, of why this is coming. So in our paper we've provided, we've had some calculations done to try and understand where this comes from, and it's essentially a negative coding, so that most amino acids like to fold back and bind to all RNAs, and that in some instances the amino acids are in a conformation where they can transfer. We're not entirely satisfied with that explanation because it's a very messy answer, but we don't have a better answer for it at the moment. I think we haven't looked much at buffer conditions. We've got it working, and we've kind of stuck with what we've worked with, and we're usually not so super interested in kind of scoping out, oh, what happens if you change salt by a factor of 2 or something like that. Once we get things working, and provided it's not ridiculously improbatically plausible, we kind of stick with that. But there must be some kind of mechanism by which RNA is mediating it. Hopefully that answers your question.
So Professor Braun asks, do I get this correctly that in the JAX 2022 by you, the activation step was under EDC, but does it then not wonder that you do not find a chirality signature since you anyhow force it?
Interestingly, with that, so yes, we do need EDC, and unfortunately, it's one of the failures of prebiotic chemistry at the moment is activation is a long-standing problem that has been.
Would you mind explaining for the audience what EDC is? I don't think
yes,
that might not be something that everyone knows.
No, that's fine. So EDC is a chemical reagent widely used for making peptide bonds, so the bonds between amino acids, which produce yeah, peptides. And it's not prebiotically plausible. It's a model molecule essentially, and this is because essentially we have there isn't nobody's presented a decent activation strategy that's kind of universal like this. What's interesting is that we found that without imidazole, the reaction doesn't occur. So, and the most likely, our most likely ideas behind that is that essentially EDC is allowing us to access the aminoacyl imidazolide. So, essentially, I don't have a good diagram to show you, but essentially, it's something that I think we could consider more prebiotically plausible because it's closer to, how like Jack, for instance, does his RNA replication chemistry and so on. But yeah, no, you're totally right to call that the EDC. The chirality thing, wonder that you do not find a chirality signature. So we do see a chirality signature with both chemistries. Eh, and they're both the same. They both like the, natural RNA likes natural amino acids. But I think when it comes to the coding, it's an interesting question about whether I'd be surprised because the yields we get between the two chemistries are very comparable. Think like the best mixed anhydride yield we got was something like 60%, and then the best amidate ester chemistry yield we got was also 60%. So that doesn't sound like forcing to me, the relative. But I understand the point that essentially if you have something that's very reactive, you can lose selectivity essentially. So yeah, interesting point, Dieter.
So on my screen, I have two hands up. Kevin is first. So Kevin, if you want to go ahead and unmute yourself and ask your question.
Okay, yes. Thanks very much, Sam. Very interesting talk. You mentioned about the problem of acquiring polymeric RNA
Mm.
prebiotically. Are you familiar with the paper? You must be familiar with the papers that came out of the Benner lab in the last year, in the last 18 months about if you have the RNA to triphosphates, which can also be made prebiotically, if you treat them on the sterile prebiotic glasses, like obtained from volcanic ashes, you can actually make appreciable amounts of RNA.
Yes, I'm familiar with this paper. I'm so so I remember seeing some of the gels from that paper, and there's some things that kind of confuse me because there are components that appear and then disappear. When from the zero time point to they're in the zero time point and then they disappear and then they come back in the final products. And then I'm also a little bit confused about the some of the resolution that you'll get that's being got by those experiments. And so I'd like if yeah I haven't had a chance to speak to the authors about that to clarify that, and I couldn't speak to the plausibility of the glasses, but yeah, I am aware of it. But I think there's still room for more work to be
I mean,
done.
the actual they were actually determined. The actual and the majority were in the naturally occurring 5' to 3' sort of
Mm,
direction. There were some 5' 2'. There were indeed. How they were identified using traditional methods that were used to actually determine the structure of RNA nucleotides developed in Alexander Todd's laboratory back in the 1950s. So I
yeah.
think the actual analysis is pretty spot on. I think it is. So
Okay.
obviously more needs to be done. I mean, the work that came out of your previous supervisor John Sutherland's lab was very, very good. I really do like it. I think it's very elegant, and it explains the sort of like prebiotic origins of all the major amino acids, all the 20 proteinogenic amino acids. Got one quick question for you. You say about naturally occurring RNA, dsRNA, D-ribose RNA,
Yeah.
as a preference for L amino acids. Is
Mm.
that Have you tried the mirror image experiment with unnatural L ribose RNA? Is there a preference for D amino acids?
Yeah, I would love to, but it's extremely expensive
And a
to do
question
it
of
on the
time and a
scale.
question of time and yeah, okay.
Yeah, so we wouldn't anticipate there would be, I would anticipate that, yeah, you would see the reverse.
Right.
Yeah,
Yeah,
so the relationship would still hold. But there is some really heroic work. There's a group in China who are like synthesizing opposite chirality polymerases and things like this to be able to make all this stuff. And I mean,
right.
reading their SI is just like, oh my God, I
Yeah.
would not want to do this. But it's and they're showing the same thing, that essentially all the essentially the mirror images can't crosstalk, and but that all everything, all the rates are the same. Everything behaves the same in the mirror image as you would expect.
Yeah, indeed. Okay. Thank you.
Thanks, Kevin.
I think so. We have one more hand raised and then a few questions in the chat. So we'll go to the hand raised first, and that's Furkan. You can go ahead and unmute yourself.
Hi, Furkan.
Good. Can you hear me?
Yeah.
Yeah, Sam, brilliant talk as always. I actually want to like kind of follow up on Kevin's point, and also I think it would answer Dieter's question partially as well. So in Jack's lab, like they actually have tried recently using L-tRNA analogs to make D peptides, and they observed the mirror like image relationship with like same stir selectivity with leucine, lysine, and alanine, I think. Which actually probably answers Dieter's question, because although the activation agent's chirality doesn't change, just by changing the sugar's chirality, they observe the opposite relationship. Maybe Sam can elaborate more on that if you're up to date with their work, and then I can ask my questions as well.
It's not something I was aware of actually. I mean, the EDC itself isn't chiral either. Like so you don't have to have like just like a geometric shape doesn't have doesn't have to be chiral.
Right.
So and in this case EDC isn't, so that shouldn't affect the result. So it's not surprising that this is the first I'm hearing of it, and it's great that they've tried it. But yeah, I think it's not surprising that the selectivity is the same, or is the mirror.
Exactly. They observed the opposite relationship with like 3 more amino acids as well, which is actually quite cool. So like the questions that I had was like I would like to hear your opinion about like purines, in the sense that like if like one day, I mean, we will figure out how to make purines, you will figure it out. And if it doesn't come from RAO, and let's say it is not produced in a homochiral way, do you think it would matter?
So you're invoking a scenario where you've kind of got an orthogonal route to the purines and you're making both L and D. So you end up with polymers that are, say, clean in the pyrimidines, so that's C and U, and they are one chirality. But so it's essentially like a 6-base system, but it's not the bases that are changing, it's the backbone.
Correct.
So I think you're going to have, my first reaction is your replication is going to be really hard, because your backbone is not going to be like this regimented system that kind of forms nice helixes. It's going to be flip-flopping all over the place. And when you do that replication, because I don't know, because you've got this structural effect, maybe you could actually get fidelity. But my first thought is that when you have, so replication depends on hydrogen bonding. So we've got the figure here where you've got these different oxygens and nitrogens, and these are what determine the identity of the bases, essentially, because having those hydrogen bonds dictate what other bases it can bind to. But if you have a system where there's essentially 2 Gs that have the same hydrogen bonding pattern, you can't distinguish between those. But then I was second guessing, because then you also have a structural feature where the bases are going to be in different orientations, so maybe there would be a way around it. My initial reaction is sounds horrendous. But yeah, it would perhaps be interesting. I wouldn't be looking into it unless I had a really good synthesis of the purines that were unchirally selective.
Right. Like perhaps like in the dinucleotide formation, like Jack is doing, there would be some selectivity that is imposed by pyrimidines on purines if they are, if they're heterochiral. But that's obviously speculation.
Yeah. No, I could totally see that, but it also depends on the template, right? Because if it would be true if your template is homochiral, as modern biology is, and so maybe there would be a selection pressure because it'd be more like homogeneous or something. But in the scenario where you kind of have equal of both, it'd be like the amount of dead material is certainly not chemically satisfying. I mean, maybe it's plausible to go around it, but as a chemist, it doesn't sound, it sounds very inefficient. But I realize that that's my bias, as I'm speaking. So
yeah, and the other question I had was about like in your aminoacylation work and in the previous works by Longfei as well. Like you study aminoacylation with various different amino acids, but you look at the stereoselectivity of alanine only. Is there a reason for that, or is it just you didn't bother checking the others?
I can't speak for Longfei. I know his paper, they spent more time on so they looked at different sequences, whereas my work I actually did do more than just alanine. I've lost it now. But anyway, just show the figure just to prove it. Let's try showing again. So you can see here that I've done L and D for alanine, valine, leucine, and serine, and leucine also in a Nick duplex context. So I've done it for several cases. I can't speak for why, but in Longfei's original paper, alanine is the only. I think they only do alanine and glycine anyway in that original paper.
I see. And what I can't really see it, but like what is the stereoselectivity for others? Like, is it on the same order?
Yeah. So the selectivity in Longfei's one is like what's that? That's like an order of magnitude.
Yeah.
My one, it varies from case to case, and actually also time points, because if you leave mine long enough, the L starts to top out just because of it's there's not that much amino acid, and then the D does start to very slowly tick over. So it kind of depends what time point you take, but yeah, you can get an order of magnitude, I'd say.
Right. So there is like a semi-universal like relationship.
Yes, there are going to be sequences that that's not the case for, as shown by the sequencing. And with the sequencing, we only looked at L and D alanine, but that's just the practicality of the experiment issue.
All right, that's great. Thank you. Thank you so much.
Yeah, great questions, Svekan. Thank you.
Thank you. So we have, I think, 2 more questions in the chat. So the first one from Roberto is, can Sam elaborate on why having a peptide joined to a long RNA as opposed to a short one leads to RNA hydrolysis? I'm not sure if I understand that statement correctly. I don't want to put words in Sam's mouth, but I don't think that's what he was saying. I think he was saying that the 2 esters happen to hydrolyze at the same rate. Is that correct, or roughly?
Yeah. So essentially, let's get that back up so people know what we're talking about. So essentially, in this scenario, you could hydrolyze this bond, right, which would then reform this and allow you to do the activation. But this isn't the only base labile bond here, right? So all between each of the RNA nucleotides, you've got this phosphodiester, which is vulnerable to base. So the base picks off this hydrogen, and then the negative charge hits the phosphate, makes a 2,3 cyclic phosphate, kicks out this OH as a leaving group, and then you get strand cleavage. So essentially, you end up starting from a longer sequence and then shortening it. And for longer sequences, because you have more of these phosphodiester bonds, then essentially the rate that the chance of per second, the chance of one of these bonds breaking increases the longer the RNA is, because there are more of them. And so I was invoking essentially, so even though this bond is more base labile generally, because there is only one of them, when you get to say like a 50-mer, we haven't got any published data, but just some very kind of quick and dirty experiments we've done in the lab suggest that kind of 50 to 70 mer RNA, you're actually on the order of magnitude of some of these of the hydrolysis rate of some of these amino acids. So essentially, for every for at that kind of length, for every if you have one of the long-lived amino acids, if you cleave for every one of these that you cleave, you're also breaking an RNA as well. So then you're losing, every time you do this deprotection, you're losing 50% of your RNAs. Which I think would be enough to provide an evolutionary driving force, right, to find a better way of doing it. I mean, this scenario, the driving force could just simply be a ribozyme that does that deacylation more effectively, and that would be another way of doing it. But we're thinking about how you get, not about whether this is real. We're thinking about scenarios that let you get to transpeptidation.
So one final question in the chat. Nilufar says, great talk, Sam. For biologically absent peptide sequences, nullimers, do you think that the tRNA chemistry and preference might play a role in not selecting them? I'm unaware of the possibility, but would like to get some input.
Biologically absent peptide sequences. So essentially, sequences are like possible peptide sequences that biology doesn't use for anything, I think is the way that I'm reading.
That's how I'm interpreting it as well. Yeah.
Do you think tRNA chemistry and preference might play a role in not selecting them? I'm unaware of the possibility. We'd like to get some input. So tRNAs are like it's like the paint palette essentially of options. So in biology, you have all the amino acids are possible. Where the selection of which amino acid of the order of amino acid comes is from, or at least the first level of selection for it, I should say, is in the genetic code itself, right? So it's about how the so it's about essentially the sequence of bases that are in the genetic code. So thinking on my feet, the peptide sequences that aren't biologically relevant would be dependent on those genes, or on that genetic sequence not existing. There's going to be at least at the origin of life, there's also going to be the added selection of this data that I was talking about. There's currently on this one's on bioRxiv. You can go look for it, about which codons have strong interactions with single-stranded RNA, and so essentially which codons and thus which amino acids would have been important for the very first forms of like this coded peptide synthesis. So that would have been an additional layer on this. But I'm not sure the selectivity would have come from the tRNAs. I think that beyond that kind of selectivity, where which ones would have been able to bind, I think that it's about where what sequences of the genetic code you can build. And I think that's a super interesting question, right? And it's why I find this oligomerization idea really interesting, because if your oligomerization biases towards certain sequences, then that will influence what RNAs you can get to and what functions you can get to as a result, right? So over lockdown, I swear I read this paper, but I cannot find it, or this idea, but I cannot find this anywhere since I've looked for it. I'm fairly sure it was Orgal, but anyway, that essentially when you have an elongating strand, at some point it's going to interact with itself. It's going to be long enough that it can fold back and interact with itself. And when it does that, it's going to want to self-prime and extend and do templated things. And then that builds a hairpin, right? And so you have this now hairpin loop with a double strand. And if you keep doing that, that's going to run off and then become random again and then interact with itself. So you can start to make in theory, you could start to make poly hairpins if you could do that. And I think it's interesting, because then you contrast that with Dieter's work that's shown that actually there isn't this kind of selectivity, or at least so far as I can tell, nobody's been able to spot that you can make this kind of, or that there's like structural sequence specificity, I don't think, but maybe Dieter can speak to that a little bit more. But interesting questions, but I think maybe tRNAs are the wrong place to look.
So Dieter just said, no, that we see so far. Yeah.
Yeah. I mean, maybe it's a length thing as well, maybe, but I don't know.
So I don't think there are any more questions. So I'd like to thank Sam again for giving an absolutely wonderful presentation for our inaugural session of the 2024 seminar series. So thank you, Sam. And thank you, everyone, for attending, and thank you all for the great questions. And please be on the lookout for our notifications about the April seminar. So yeah, thanks very much. Take care.
Thank you very much. Have a good one, guys.